Thank you. It's my very great honor to introduce Dr. Nicholas Lydon, who is to receive the Hope Funds for Cancer Research Award for Clinical Development. Dr. Lydon received his Ph.D. in biochemistry from the University of Dundee and is a fellow of the Royal Society. Thanks to his decision to become a biochemist, not only have hundreds of thousands of patients survived cancer, but the whole approach to treating cancer has been transformed. His work discovering and developing protein kinase inhibitors resulted in the development of Gleevec, for which he and two colleagues were awarded the Lasker Prize, among other awards. Gleevec was more than just a breakthrough treatment for chronic myelogenous leukemia, CML as it's known, and other cancers. It ushered in a new area of cancer treatments. Gleevec binds to and inactivates BCR-ABL, as you'll hear about, a mutant overactive tyrosine kinase protein responsible for cancer cells wildly reproducing. This was not only the first drug to target a specific molecular biochemical mechanism to shut down the multiplication of tumor cells, but on top of that, it was an oral medication. The clinical success of Gleevec was a catalyst for the whole field of cancer drug development. Dr. Lydon has also proven to be a leader in inspiration for cancer research by the way he's performed his research, including his partnership with his academic and clinical collaborators and his pioneering of new companies, and I may say successful companies. Dr. Lydon, for your pioneering work in the clinical development of Gleevec, which helped lead the way for the successful development and approval of other targeted therapies, and your devotion to improving the life of cancer patients, the Hope Funds for Cancer Research is delighted to present you with this award for excellence in clinical development. Dr. Lydon.
Thank you, Robert, for the very kind words and introduction. And thank you, the benefactors here supporting the Hope Funds in their incredibly important work. So when Leah informed me I'd been selected for this prestigious award, it made me think back to when I was a young student starting my career in oncology research. And some of the key contributing factors I came up with was firstly luck. And in my case, that involved being at the right place at the right time, which has been incredibly important all through my career. The second were my mentors, and they really helped me build a strong foundation early on in my career, which I've been able to build on and is incredibly important for those of you mentoring students to build that foundation early. And then the third was the collaborators I've built relationships with, and many of those I started in my early career I still work with. For example, Brian Drucker and George Dimitri, who have been prior honorees receiving this award. So I'll take you back to how I started my career, and that journey started in the late 70s when I decided to apply for a graduate position at the University of Dundee. And I really had no clue what graduate training was, research training. And I ended up in a small lab in Dundee as the only student, the only researcher working on one of the first protein kinases, protein kinase A, and the hormonal regulation of protein kinase A. And this was obviously not a very good environment to learn how to do research. But luckily for me, one of my big breaks, in the lab opposite, Sir Philip Cohen's group was working, and they were the pioneering group in protein kinase research, looking at the regulation of glucose glycogen storage and metabolism, and working out the signaling pathways, whereas reversible phosphorylation regulated this important signaling pathway. And I was really struck by these molecular switches, these protein kinases, which would switch on and off pathways to regulate complex biological problems. So I was incredibly lucky because the postdocs in Philip Cohen's lab really took me under their wing and helped me to graduate. And without their help, I probably wouldn't have continued in science. So I moved into pharmaceutical research in the late 80s under the mentorship of Alex Matter, who had started up a oncology group at Ciba Geigy, which then became Novartis. And that era witnessed the sort of convergence of work on protein kinases, oncogenes, retroviral oncogenes, and cancer pathogenesis. And it was an incredibly interesting time. And based on my undergraduate and postgraduate training, I was highly sensitized to that relationship between deregulated protein kinases and cancer pathogenesis. So I was highly primed to take advantage of the emerging work coming out of the oncogene field. So based on the work of Janet Rowley, which I think some of you have heard today at the lunchtime talk, the Philadelphia chromosome and BCR-Abelson fusion protein were really the only truly validated human oncogenes at that time. Not oncogenes, kinase oncogenes. And so they made the very obvious target to start my work. So with the mentorship and trust of Alex Matter, I started my first drug discovery project targeting the BCR-Abelson oncogene. So it was a pretty good target to get started on my career. So I have to mention Peter Traxler. He was the head of medicinal chemistry working on my project at Ciba Geigy, and he was really a mentor who taught me all the foundational things I've relied on in how to discover and develop drugs. And without him, I would have found it as an inexperienced biochemist almost impossible to navigate the path to develop a drug at that early stage in my career. I also have to mention Brian Hemmings. Brian was a postdoc in the Cohen lab who had moved to the Friedrich Miescher Institute in Basel a little bit before me, expert in kinases, and he really was a sounding board for all my ideas and really helped me get the Abelson project off the ground and introduced me to a lot of molecular techniques that my lab learned to get me started in the field when I was joining Ciba Geigy. So as many of you know, as the project progressed, when I started working on Abelson, there were only a handful of known protein kinases. But as the project progressed, new protein kinases arrived out of the woodwork on a daily basis. And by the time the project was mature, there were over 400 known protein kinases from the sequencing data. So this made it really much more challenging project to come up with selective drugs that targeted this particular oncogene. So in the early 90s, I started a collaboration with Tom Roberts at the Dana-Farber Cancer Institute to enable us to make a panel of recombinant protein kinases to monitor selectivity. And Tom had pioneered some early work techniques using baculovirus expression to make these enzymes, and I collaborated with him to establish the panel of enzymes we relied on during my discovery work. And it was during that time I met Brian Drucker, who has been mentioned earlier and is a prior honoree. Brian was a clinical investigator in Tom's lab, and that was how I met Brian. So skip forward to around about 93, along with my key collaborators at Ciba Geigy. There was Elisabeth Buchtunger and Jörg Zimmermann. We finally identified the Abelson KIT inhibitor, imatinib. By then, Brian had moved to the OHSU in Oregon, in Portland, Oregon, and I started an immensely productive translational collaboration with Brian, looking at Gleevec in his cell models, and most importantly, in ex vivo patient samples from some of his patients. And this really convinced us that imatinib would work, and Brian subsequently became the principal investigator for the clinical development of imatinib and presented the really exciting early results at ASH in '99, which really laid the way for Gleevec to be approved as one of the first targeted therapy drugs in oncology. So since that time, since the success of imatinib in CML, and also due to the work of another of my collaborators, George Dimitri, in GIST, I've continued to work on especially the KIT oncogene-driven pathway. And two examples of that are a Blueprint Medicine, where we've recently developed avapritinib and elenatinib for mast cell-driven tumors and also for indolent systemic mastocytosis. And more recently, in collaboration with George Dimitri, we are developing IDRX-42, which we've just presented at ASCO, which is showing real promise in treating, I think, second-line drug-resistant GIST. So to finish up, as many has said before me, successful drug development is really a team sport, and you really can't claim credit for developing a drug. It really requires a large array of skills, and I've been very fortunate to work with project teams over my team who have time in pharmaceutical development who have really delivered on a number of projects. And I would love to accept this award on behalf of those project teams. Thank you very much.